Okay, back again in a Mammalian Histology course, UIBSC Zoology, paper 2. Here we'll see syllabus point 3.9 today, a B core B point, that is a ovary, a female reproductive system part. Here we'll give more emphasis on primary follicles, secondary follicles, and Graafian follicles. Also, we'll see detailed histological structures of corpus luteum and corpus albicans. Here, uh, in this slide, we are showing this female reproductive system, entire female reproductive system. Among these, we are interested for this course in histology of these ovaries only. These are the two pair structures present in abdominal cavity. They are hanging in abdominal cavity and attached with these ovarian ligaments. Over to each ovary, there is a fallopian tube which receives ovum and place it into the uterus where it gets fertilized and develop further. So for this next few slides, we will confine ourselves for the histology of ovary only. Here it is the actual arrangement of ovary in an abdominal cavity. They hang like this in an abdominal cavity with these fallopian tubes. Here in a cross section, they have shown how the ovary is attached in abdominal cavity and it is related with the other reproductive organs in our female reproductive system. This is the uterus, the fallopian tube. They receive their eggs and transferred and get implanted in this uterine wall. Now, this ovarian tissue, if you remember that tunica albugia thing we already discussed in a testis, the outer layer of a testis, that is of a tunica albugia. The same tunica albugia forms the outer layer of ovary here. But internally, all these albugia is divided into a medullary and cortex regions. Depending on presence of some specialized tissues, this entire tunica albugia matrix is divided into two parts, as a medullary portion, the central portion, and the cortical portion, that is surrounded portions. So in a cortex, the cortex is composed of the ovarian follicles. Ovarian follicle, this is a new word for you, maybe. Ovary is a site of where there is a cytogenesis occurs. That means there is a germinal layer and they forms the oocytes. After oogenesis, from the germinal layer there develops the oocytes. So that way that ovaries are cytogenic glands, as well as what they are also endocrine glands because they produce hormone estrogen and progesterone. And this effect of hormones then control all the development of other reproductive organs in a female body. So this cortex is composed of these ovarian follicles at different stages of development of oocyte, that is called as a follicle, ovarian follicles. They are surrounded and scattered into this connective tissue matrix in the cortex portions. These follicles are spatial structures that are each composed of somatic cells called as a follicle cells that are surrounded by oocyte. This oocyte is a germinal cell and they are supported with the normal cells, that is somatic cells, and they together form this follicle. In addition to the spindle-shaped fibroblast, the stroma of cortex also contains collagen and reticular fibers. This is but natural to provide the framework and to provide the attachment and substance for holding this developing ovarian follicle. Ovary required some support of connective tissue like fibroblast and collagen fibers, reticular fibers, which forms the matrix or also base in which these follicles are developing. Now in medulla portion, that is the core portion, central portions, there are blood vessels and now entering into the ovary and they provide nourishment to the cortex portions, and overall that ovary nourishment is provided with the blood vessels. So that helical arteries and veins you will see in the middle portions. There also you will find some nerves in a dense stroma of supporting tissues. So all that connective tissue are holding these nerves and the blood vessels. These nerves and blood vessels enter and leave ovary through the hilus of connective tissue. It's an opening kind of a structure present in ovary. We'll see that in the next few slides. There is an opening like structure in the ovary, and that structure is attached with the ovarian ligament. If you remember the first slide, there is a ovarian ligament. Through that ligament only, these blood vessels, nerves, they enter into the connectivity, enter into the ovary. That portion is called as a hilus. Now, this is a diagram, and many times in university exam they ask draw the diagram of ovary indicating the histological structure. So that time you have to draw this diagram. So it is presented here in a diagrammatic fashion. Now all what we see in the first two slides is revised here. So this is a ligament portion which is attached with the main body. So through this ligament, all that blood vessels and nerves enter in the ovary. So this is called as a hilus portion. This is a connective tissue portion that is called hilus. So that earlier sentence, that blood supply and nerves enter through the hilus into the ovary, that fulfill here. Now all these ovary, they are covered with a tunica albuginea. This is a layer of tunica albuginea which covers entire ovary. The same tunica albuginea we have seen covering around the testes in the earlier lecture. So that tunica albuginea is there, and immediately after tunica albuginea, there you will find there is a germinal epithelial layer is there. Germinal epithelial layer from which the germ gametes or germ cells develops, and they form the, in case of ovary, they form here ovum. In case of testes, we have seen that the germinal layer gives spermatocytes, spermatogonia, and that spermatozoa. So here, that germinal layer, they produce oocytes. Now, apart from this cortex portion here, this is all cortex portions, which shows a very minute development of these germ cells. Now they are called the primordial follicles. In the as it goes into the middle portion here, in the medulla portions, you will find there is a lot of blood vessels. All these red color dot and structures with red color dot, these are the blood vessels. These indicate the blood vessels, and many times you will see here these black color structures, they are nerve endings. Why there are so much nerve endings are there? Because these ovaries are not only an organ for producing the ovum, but it also a kind of endocrine organ. Okay, so apart from this, we'll see some other labels also here, which we will discuss later on. So we start with this. So these are the primordial follicles, then these primary follicles, then these are the secondary follicles, then here is a corpus luteum, then corpus albicans, and this is a Garfian follicle. Now if you want to read this in a sequence, the sequence is primordial follicle. That is the first stage of development of germinal epithelial cells. Thereafter primordial follicle, you can see the development of primary follicle. Okay, thereafter you see here development of these secondary follicles. After secondary follicle, the tertiary follicle, or it is also called as a Graafian follicle. Now this is a final structure of a follicle which is now ready to eject out ovum. So in centrally located here, you can see this is a developing ovum, and when this is called as a oocyte, but when this border line gets ruptured and they release that ovum outside this ovary, that oocyte is termed as a ovum then. What happen after ejaculation or after ejecting out this oocyte with this graphene follicle? That graphene follicle become later on change its histology, and now it is a uh, endocrine organ, and so it is called as a corpus luteum that goes on producing progesterone. Now, after, if that pregnancy sustained, this corpus luteum go on continually producing progesterone till the end of pregnancy. After pregnancy, or if pregnancy doesn't happen, then this corpus luteum get degenerated, and thereafter it is called as a corpus albicans. And after degeneration of this corpus albicans, now it is called the atretic follicles. So that is end of this follicular journey. Earlier only we told about what is follicle. Follicle is a supporting somatic cells to the germinal epithelial or germinal oocytes. So it's not only the oocyte, it is a collection of somatic cells along with the germinal cells. Okay, now in this color photograph of cross sections of ovary, that is ovaries of rat. Okay. So here mammal, it's a ovaries of mammal and the cross section of ovary. Now here we will verify that diagrammatic labeling and structures here again. So see, this is a opening portion of ovary. So this is, you can say, it is a hilus portion through which that the ligaments are attached to the body, and through that ligaments will support only, this is a made up of all connective tissues, and through this there enters all that blood supply, and the nerves ending enter through this portion hilus into the medulla portion. Okay, so this is the medulla portion, and the surrounding border area is the cortex portion. Now what else you can see here is all these vacuoles of some small and large size. They have different size vacuole you can see here. All these vacuoles indicate what they are, the developing follicles that may be primordial, primary, secondary, tertiary, or graphene follicles. Or see here, it is irregular shaped structure, and this is a corpus luteum. Okay, it is a functional endocrine organ in the ovary. After ejaculating or after expelling out that oocytes, ovum, from the ovary, this is a corpus luteum structure. So I think this diagram, this color photograph corresponds with our earlier diagrammatic presentation. So that diagram we can use for drawing in our university exam as a diagrammatic presentation of ovary. So here again we can see the presence of all that follicles. This is again the cross section of ovary. Now here they have shown very clearly developing primordial follicles. This is a follicle. Again, this is a small primary, secondary follicles. This is a corpus luteum. Now we'll start with the primordial follicle. It's a one incomplete layer of latent follicle cells that surround the central oocyte or immature egg. In this previous slide only we have seen a vacuole-like structure. So whatever the cells are lying internally to that follicle, these are what is the primary follicle or flattened follicular cells. These follicles are located just beneath the tunica albugia and they form groups of primordial follicles called egg nest. Now in mammals, the number of oocytes, or you can say the germinal epithelial layer, how many ovums that ovary will produce, that is fixed and definite. Throughout the life, they go on producing that oocytes, and at the one end and the one moment, all that oocyte gets exhausted, and thereafter there is no development of a oocyte, and so there is no development of follicle cells happens. Okay, so the oocyte diameter is about 40 micron in diameter. Now this is how they look, primordial follicles. So this is the outer portion, that is cortex portion of the ovary, and here they have shown this primordial follicle. Now number of these primordial follicles containing the nucleus ovum is a fixed. This number of primordial follicles in a human body, their number is a fixed. And so all these supporting nourishing cells are there to this ovum. So central portion is the nucleus and surrounding is the follicular cells. Now as the development goes on, slowly these follicular cells get functional and they start enlarging. The number also goes on increasing and thereafter they are called as the stromal cells. So here you can see the primary follicle, the central this portion is a ovum and all these surrounding are the follicular cells. The primary follicles, these are the follicles that have begun to mature at the beginning of most recent menstrual cycle. Okay, we forget to talk about the menstrual cycle. These ovarian hormones produce a cyclical change in a female reproductive system, which is responsible for the continuous change, histological change in a endometrium, that is called as the uterus, internal lining of uterus goes on changing cyclically, that is called as a menstrual cycle. So these are the primary follicles that have begun to mature at the beginning of the most recent menstrual cycle. So this maturity of primary follicles from primary to secondary, primordial to primary, then primary to secondary, tertiary, vacuum follicle, corpus luteum, all these events happening in a ovary, they are very much synchronized with the menstrual cycle, stages of menstrual cycle. So as the menstrual cycle goes on advancing, here there is a goes on maturations of a follicles that consist of 1 to 7 layers of cuboidal follicular cells that surround the central oocyte. We have just seen in a previous slide many cuboidal cells surrounding the oocyte. There are no fluid space between the follicular cells. So till in a primary follicle state, that central portion, the hollow portions around the oocyte and the follicular cell, it is empty. During the primary follicle stage, the oocyte grows to about 110 micrometer in diameter. It develops a thick acidular layers of glycoprotein called as a zona pellucida that completely surrounds the oocytes. Now we don't go in much detail about the size of the ovum in a human because we are learning mammalian histology, and with respect to different mammals, this size varies. So they have quite good enlarged size of a oocyte. That much is enough for mammalian histology. Now apart from these follicular cells around the oocyte in a primary follicle, what else is there? It is the glycoproteins. A zone or layer of a glycoproteins, a cellular layer of a glycoprotein that is called as zona pellucida. Now the plasmolema of oocytes and the adjacent plasmolema of the adjacent layer of follicle cells develop many microvilli that extend into zona pellucida and interdigitate with one another. Now if you carefully see these primary follicles, again I will show you that slide. There we have to verify all those points which are mentioned here. So what happens that follicular cells produce a microvilli extending to the central portions of the primary follicle towards the oocytes, and this glycoprotein layer get connected with the with these follicular cells microvilli, and they form the connection between them. This interdigitations of microvilli increases the surface area of contact between oocytes and its adjacent follicular cell layer. So slowly these oocytes internally go on touching the follicular cells and increasing its contact with the oocytes. That way there is a more exchange of nutrient take place to the oocytes. So we can say that follicular cells play a role of nourishing the developing oocyte. Do you remember what is the role of Sertoli cell in case of testes with respect to sperm? That nutrition is provided to the sperm through the Sertoli cells. So same way here, these follicular cells take care of the nutrition of oocytes. This the area over which nutrients can pass. This is what they have written again here. For nourishing and for collecting the waste from the oocyte, that follicular cells play a role. The layer of follicle cells that is directly adjacent to the zona pellucida is called as now corona radiata. Okay, one more terminology added here. So around the oocyte, whatever the zona pellucida form and whatever the microvilli connection between the follicular cells and the oocyte happen, that portion is called as a corona radiata. Theca interna forms from the stromal cells that surround the original follicular cell layer. Again, that stromal cells, the developed follicular cells now called as stromal cells that they form a connection to the that follicular layer that is called as theca interna. Okay, so here now whatever we see in a first slide that we have to verify with this color photograph. Now for drawing purpose, we have provided here the diagrammatic presentation. These are the actual photograph of a slide. Okay, so first we'll see diagrammatic presentation here. So this is the oocyte. Okay, and all these zona granulosa. This is a zona pellucida layer, and this is a theca externa portion and theca interna portion. Okay, so this zona pellucida is a layer of a glycoprotein. Zona granulosa is a layer of a follicular cells. Then zona, this theca externa and theca interna is a formed because of the stromal cells, and centrally located is a oocyte. Okay, so we'll verify this in this two color photograph. So here it is a oocyte and the nucleus of a oocyte. And what this all is filled with what? This is a zona pellucida. This is zona granulosa portion, a follicular cells portion. And outside this is a theca interna and theca externa. So here again, this is a primary follicle, this is a primordial follicle. Okay, so here there is a formation of these two layers, zona granulosa and zona pellucida, happen in a primary follicle, which are absent in a primordial follicle. In primordial follicle, you will not see this glycoprotein layer and this zona granulosa and this theca externa. In that case, there is only a stromal cells are there and the oocytes and surrounding follicular cells. But as they go on developing, they form this new terminology there. This is a electromicrograph of a follicles, that is a follicle in a ovary, to confirm whether there is really a zona pellucida follicular cells are there or not. Okay, so this portion is a nucleus, ovum, and this is nucleus of ovum. So here it is all mitochondria in a ovum, and these are the glycoprotein layer, zona pellucida portions, and these are the follicular cells. Okay, and so basement membrane outside to this, there you will find the theca interna and thereafter theca externa layer. So yes, that confirm the presence of all what we learned